Hello, everyone, and thank you for joining us. A couple of housekeeping remarks before we begin. We do take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the Q&A tab on your dashboard at any time, and if you need technical assistance, please use the chat tab. So at this point, I'd like to introduce our speaker, Joyce McWan, Senior Scientist at Simulations Plus, and she's going to be discussing the use of PBBM to establish dissolution safe space via IVIVR for Concor.
Hello, everyone. Thank you for taking time to join this session. So today I'll be briefly describing the work that we published last year with Mark, and we'll be discussing how we use the PBPM and virtual BE approach to establish the dissolution safe space for their product Concor. So as an internal quality system, Merck tests the dissolution on a limited batches for their Concor product. And they observed the slight differences over the years for different batches. So some of the batches, they showed fast dissolving profile, and the other batches, they showed the slow dissolution profile. So what we did, we used this PBPM and the virtual B approach in order to evaluate whether these differences in dissolution of Concor would impact the simulated PK of bisoprolol. And we build the IVIVR using the in vitro dissolution data and available plasma data for the bisoprolol, and then we assess whether there is a significant impact or not on the clinical performance through the PBPM and the virtual B approach. So we use the GastroPlus version 9.6 to build this PBPK model. We used our mechanistic ACAT absorption model in order to include the in vivo dissolution and the intestinal absorption of the compound. And we use the full PBPK model to describe the distributions and the elimination of this compound. So as you might know, in PBPK or PBPM model, we need to provide the physicochemical and biopharmaceutical properties of the compound. So we use the mostly the in vitro data because this compound is very well characterized. So we could find in vitro data in the literature. And we also obtained some parameter value from the Merck as well. In the absence of experimental data, we relied on the in silico values, and we obtained the in silico estimates from our ADMET predictor software, which is based on the chemical structure of the compound. And when necessary, we fitted the value of certain parameters in the model. So in order to build the underlying model, based underlying PPK model, we mostly rely on the literature data. And for the literature data, we use this pair physiology in order to create the population. So we gather the information on the mean age, mean body weight, and the race of the subjects, and we created that population from getting the information from the literature. We treated all the tissues as a perfusion limited tissue, and we calculated the tissue to plasma partition coefficient using our default Luka-Chova method. And we obtained the IV data in the literature. That was we used to calibrate the volume of distribution method in the PBPM model. And I just want to tell you some important properties of the compound. So this compound has a high log P and high base pKa. And we have learned from the literature that the compound, which has a high log P and high base pKa, they tend to undergo lysosomal trapping. And as a result, this compound usually have a higher volume of distribution, as well as they also show delay in the Tmax. So in order to account the effect of lysosomal trapping on volume of distribution, we increase the blood to plasma ratio to 1.1 value. And also we decrease the enterocyte default value from 100 to 5% to explain the delay in Tmax and to include the impact of lysosomal trapping in the enterocyte. This compound is equally clear by both metabolic and renal excretion, so we included both mechanisms in the model. We did find some in vitro clearance data in the literature, so that was we plugged in the model, but which was not predictive of in vivo. So we ended up scaling that in vitro clearance in order to match the observed plasma profile from the literature. Again, from the same publication from the IV, we did find the urinary excretion data following the solution administration, and we model the renal excretion as a fraction of kidney blood flow by fitting the fraction of 0.12 in order to match the urinary excretion of unchanged bisoprolol to 50%. When it comes to the dissolution, for the baseline model, we use the Johnson dissolution model. And since we built the baseline model using the literature data, we didn't have any information on the particle size. So we just use our default particle size of 25 micrometer in order to model the in vivo dissolution of the compound. For IVIVR and virtual BE trials, we fitted the single viable function to the in vitro dissolution data. And we used the clinical batch 5080504, which was used in one of the clinical studies. So Merck actually had 4 clinical studies in which the different batch of the Concor was used. But all of these batches actually had a fast dissolving profile. So we picked up one of the clinical batch 5080504, which was used in study ALOP at 481, because this batch has a dissolution profile that falls between the fast dissolving in vitro dissolution data and the slow dissolving batches. In the absence of the clinical data for the slow batch, which is the slowest batch from the available in vitro dissolution data, batch number 231975, what we did, we first built the safe space IVIVR by predicting the CP time profile of this batch. So we use the in vitro dissolution data of this batch and assume as an in vivo input and then compare with the reference formulation. In order to build the virtual subject, we utilize the individual data from the same study and match the demographic information of the subject and created the virtual population. And that virtual population we used to assess the bioequivalence between the slow dissolution batch and the reference formulation, as well as the fast dissolution batch and reference formulation. And also we use the same population in order to expand the safe space by extrapolating the hypothetical dissolution profile beyond the knowledge space. And we'll talk more about when I discuss the results of this session. So you can see that the first 2 graphs, so first one is the figure 1a, you can see the model nicely matches the IV profile. And so we had a confidence even of our volume of distribution method. And when we applied the same model to the oral solution data, you can see the model adequately describes the observed plasma data, which is shown in the blue points, and also the urinary excretion data, which is shown in the purple line. So once we had this baseline model, we applied the same model to the several literature data sets. So we did find the several clinical data set for the single dose or multiple dose as several dose levels. I haven't shown the data here just because of the space limitation, but you can go through the publication to get the more information. So once we had the robust underlying PBPK model, we applied this model to the clinical data set of Merck as a part of a model verification. And you can see from figure C to F, the model nicely describes the observed plasma concentration time profile for bisoprolol from 4 clinical studies that was conducted by Merck. And you can see the model also matches nicely the multiple dosing data from one of the study as shown in figure D. So once we had the validated PBPM model, we used that model in order to run the several virtual BE trials. So the first step, what we did, we used our virtual study population and ran around 10 crossover trial with the slowest dissolution batch and compared the point estimates and 90% confidence interval of geometric mean for the simulated Cmax and AUC with the reference batch. I have not shown the data, but you can find in the publication, and we showed the bioequivalence between the slowest dissolution batch and the reference formulation. We did the same thing for the fast dissolving batch, and the data are shown here on the on the top row from this table, that the 90% confidence interval and point estimate for Cmax and AUC were within the Bay limits. Then what we did, we actually created 3 hypothetical dissolution profile. So what we did, we actually decreased the dissolution for the each time point, including the last time point, and we maintained the shape of the profile of those theoretical dissolution to match the shape similarity for the commonly observed dissolution profile for the bisoprolol. And we had tested 3 dissolution theoretical profile. One, and these we had 72%, 70%, 69% at 15 minutes respectively, for example 1, 2 and 3, and around 81, 79.5 and 78% at 30 minutes. Okay, and maybe I can show you here in the figure so you will get better idea. Yeah, so the last lower 3 points that you see, the blue, the green and the yellow, these are the 3 hypothetical dissolution profile. And you can see that that the example 2, which is the 70% dissolution in 15 minutes and the 69 did around 30 minutes, that was sufficient to see the bioequivalent with the reference formulation. And the yellow one, which was the 69% in the 15 minutes and 78% in 30 minutes, that was outside of a Bay limit. So that was not showing the bioequivalence with the reference formulation. So what here we are trying to demonstrate that that by doing this hypothetical dissolution profile, we wanted to show that it doesn't have to meet the requirement of the fast dissolving criteria, which is like 85% drug should be dissolved in the 15 minutes, right? We could see that even the dissolution profile, we had 70% dissolution in 50 minutes and 79.5 in 30 minutes was still bioequivalent with the reference formulation. So in conclusion, the validated PBPK model, this was used to run several crossover virtual BE trial. We demonstrated that there was no impact of fast dissolving or slow dissolving batch on the PK of the compound. We also showed the possibility that this approach can be used to request a biowaiver for a compound like bisoprolol, which is the BCS class 1 compound, which has a well-defined dissolution, well-defined absorption properties, and most importantly, because the risk for lack of in vivo performance similarity is very low. So this analysis, we also demonstrated that we could expand the safe space by extrapolating the dissolution to the beyond the knowledge space if this compound, if for the compound which have the well-defined dissolution and absorption properties. So thank you for your time, and I'm happy to take any questions you might have.
Well, thank you very much, Joyce. That was great. I don't think we have any questions just yet from the audience. We're down to about a minute. So I just wanted to remind everyone in the audience, immediately following, there is a panel discussion. So any of the speakers in this track are available for further discussion or questions if you didn't get a chance to ask during the session. So maybe at this point, we'll close out this session. Thank you everyone for joining us. The webinar is being recorded for playback and will be available on the Simulations Plus website and YouTube channel. So this will conclude our session, and thank you to Joyce and thank you to everyone for attending.
Thank you everyone. Bye-bye.